Okay, that was fun. Thanks, Brian. Our last speaker is Dr. Audrey Fan, Assistant Professor of Neurology and Biomedical Engineering and an affiliated faculty member of the Center for Neuroengineering and Medicine and the Center for Neuroscience. Dr. Fan co-leads the neuroimaging core of the Alzheimer's Disease Research Center at UC Davis Health, which is funded by the National Institute of Aging. Dr. Fan developed the National Institute of Aging. That would be fun. National Institute of Audrey. I would go. Dr. Fan develops advanced imaging methods to study human brain function and improve cerebral health across the lifespan. An important focus of her work is how the vascular supply to the brain critically supports its function, which she investigates in collaboration with engineers, neuroscientists, and stroke and dementia clinicians. Today, Dr. Fan will talk about human imaging of the vascular brain with magnetic resonance imaging. Please join me in welcoming Dr. Audrey Fan.
Thank you so much, Diaznu. Let me just pull up the slides really quick. Awesome. I am really excited to conclude today's talks today, and I'm going to share our research on magnetic resonance imaging in humans. But first, let's do an activity together, which is the Stroop test. So while you're sitting in your seats, I'd like you to read the words aloud to yourself, so the text of the words. And now tell me what color of the font these colors are. Great. Now take a look at the second line now, and let's do the same thing again. So read the words themselves. And now tell me what those colors are. Right, even for me, it's and I've practiced this, right? It's a little tougher, right? And so this is a type of neuropsychological test that can help us understand cognition, specifically things like processing speed, executive function, and attention. And you can imagine that your performance on these relative tasks might deteriorate a little bit with aging and dementia. And what I want to add on top of this is our engineering-based work, which is what is happening in the human brain as you were just doing that. And you may know that MRI is not just about creating images of your brain for structure, but it can indirectly detect your brain function. And the way it does this is through an indirect function of your blood flow. So at baseline, as you may know, our blood has hemoglobin in your red blood cells, and it helps to carry oxygen that your brain needs. When there is a task, a neuronal increase in activity, that leads to increased oxygen consumption, but even more so, your vessels dilate and there's a big delivery of blood flow. This big delivery of blood flow to the regions that your brain has activated for the task can be detected with functional MRI, fMRI. And so when you look at these images for blood oxygen level dependent fMRI, that's the visualization of the blood flow that I was just talking about. It's an indirect visualization of neuronal activity. You may have seen these blobs before as fMRI scans, and you can compare potentially the activation between younger and older people. And we typically look at these fMRI scans and we want to interpret it potentially as changes in neuronal activity, neuronal function. But for instance, this study showed that in the same people, if you can directly measure, and there are methods to measure cerebral blood flow specifically with MR, you also see similar and multiple brain regions happening, activating, and you can also compare across people. So then the question is, if you see a difference on an fMRI scan that's more qualitative, is it because the neuron activity is different, or is it because the brain's ability to supply blood flow that we detect on MRI is different? And this has additional implications when we think about networks, which is a theme of today's NeuroFest. Even when you don't have a task that necessarily you're goal-directed and focusing on, regions of the brain, not necessarily next to each other, co-fluctuate together. So you look at the MR signal of these different brain regions, they just happen even at resting state while the person is lying on the scanner to just fluctuate in sync with each other. And if we can find these regions, they are what we call functional networks with MRI. And one classic network, especially if it's late in the day and your mind is starting to wander, is called the default mode network. This default mode network actually lights up when you're not really paying attention to anything, when your mind is wandering. But you can see again that there's coordinated functional networks are brain regions in humans that serve related functions, but they could be in very different parts of the brain. And I want to point out again that these sort of resting state networks are affected by blood flow. So I'm showing here a person who has stenosis or narrowing of the vessels on one side, their connectivity is very asymmetric. And when you have these lags of signals, right, the synchrony could be affected by stenosis because the blood flow is taking longer to get there. And therefore, what you count as fluctuations in a coordinated network may fail to be detected, right? But all of this is, for me as an engineer, really interesting because it says, hey, there's information about your brain vessels and potentially about your brain vascular health embedded in these MR signals that we collect non-invasively in human brains. And I want to take advantage of that. I want to get that information. Why? Because the vascular supply of the brain is such a key piece to healthy brain aging. Our brain is only 2 to 5% of our body's mass, but it takes up to 20% of the oxygen delivered by cardiac output. And I want to acknowledge that there are devastating consequences. Some of you may know people in your families or in your friend circles who have had a stroke. Every year, nearly 800,000 people in the US alone have a stroke with devastating consequences to their health, to their families. And for those people who have strokes, 70% of stroke survivors continue on to have cognitive impairment. And longitudinal studies show that your midlife level of hypertension and diabetes is also predictive of the development of dementia as we age, right? So all key pieces from epidemiology and clinical studies that vasculature is so important to healthy brain aging. But up to now, we have, I would say, been relatively limited in our ability to detect this. So what I'm showing here, these images, especially on the right, are MRI scans. And the things that are highlighted are lesions, infarcts that may reflect a stroke, or what's called small vessel disease, affecting the tiny microvessels in your brain. But these are structural lesions. Probably a lot of things have happened already when we can see these structural changes, right? So what I want to do is be able to develop imaging that allows us to see functional changes in the vasculature before these sort of lesions even come up. And what I want to address in terms of current gaps is lack of quantification, being able to measure the vascular parameters, and I want to use it in a dynamic way, which I'll share soon about a stress test for the brain. And I also want to start to link these sort of resting state networks to our overall systemic vascular health, trying to understand what drives the vascular component of these networks that we can see on MRI. And overall, my vision with this research is to maximize the quantification that we can get out of MRI signals, especially during dynamic conditions, and with the ultimate goal of having impact toward healthy aging, understanding of mental disorders, as well as a basic foundation for our fMRI, functional MRI signals. So now I'm going to share 2 research projects in my lab. And the first is developing a so-called stress test for the brain. So just as a cardiologist may have you run on a treadmill and measure some responses to assess your cardiac health, that stress test triggers some external stimulus that the body needs to respond to, and that's an indicator of health and response that can give you information about your health beyond baseline. And the same is true for the brain. So here, this color map, the red is actually indicating responses of the brain that are able to dilate vessels in response to a stimulus in a healthy way, whereas the darker colors is where they are less able to dilate. And in order to do this, to understand what's actually happening when you give a stress test, I would argue we need to quantify. We actually need to measure it. And the way in our lab as engineers that we are trying to measure this is to rethink MRI as not just these pretty pictures, qualitative pictures, but thinking of it as a fingerprint. So if you take one part of the brain and you look at it across different time points, what we call echoes in MR, we can build a signal curve that looks like this over time. This is what we can measure on the MRI machine over time. And we're gonna call this a fingerprint and try to get from the fingerprint information about your brain vessel health. So things like, what is the volume of blood in your brain? What's the oxygen saturation, oxygenation status of your tissue? What is the vessel radius in each part of your brain? And the analogy of the fingerprint is apt in the sense that if we have a fingerprint that we can acquire, we look at particular components like loops or rings in the fingerprint, and if there's a database that we can match to, the best match will tell us something about the underlying information corresponding to that match in the library. Likewise, from our MRI scans, if we can build a fingerprint over these echoes or time points and also identify a best match to the database, then what I want to do is to be able to fingerprint these sort of vascular parameters, this underlying information from our best match fingerprint based on MRI. The key here is actually having the engineering capabilities to build this database. And that is what we have been working on in our lab. This is my student, Greg Wheeler, who just defended his thesis this week. He's in biomedical engineering, and he's really built different types of simulations with computer programs of what the blood vessels in your brain tissue might look like, vary those in a database, and say, hey, what are all the possible outcomes so that we can match and, again, get to these underlying vascular parameters. And using this technology, we as a lab have really pushed the ability to do this fingerprint matching, not only without contrast, but doing it fast, so that we can actually apply it during a brain stress test. And what I mean by a brain stress test is having people breathe just a little bit higher CO2 than what's in the normal air. That causes your vessels to dilate. We're doing this periodically, so having them breathe this for about a minute. And here are some results, for instance, from our fingerprinting technique. We're able to see these responses to the gas. So for instance, your oxygen saturation in the brain increasing with this dilation. And we want to see how this response is in magnitude, as well as how long it takes to get to that response across young and old participants. So this is our ongoing study. Our hope is with this sort of stress test, enabled by our fingerprinting measure, we can measure how our responses are in the brain, just like a cardiac stress test, and ultimately link this response to how these individuals can perform on cognitive tests like we started off our talk with. And the quantification here, the measurement, I think is key because, just as with cardiac health, there's been a lot of great clinical improvements by, for instance, understanding blood pressure and monitoring that. This might be a way to use similar metrics, measure your brain health, just like blood pressure for your heart. Now, speaking of the heart, I want to speak to a second research area in our lab, which is how the vascular brain links to the body. So now we're extending the idea of neural pathways beyond the brain into the body. And this is spearheaded by my student Quimby Lee in neuroscience, and really thinking about how our breathing and our heart rate affects functional MRI brain signals. So I showed this image before where neural activity causes your vessels in the brain to dilate, and that's how we use fMRI signals to detect function indirectly. But this vasodilation is subtly happening all the time. As you breathe, as your heart rate, like this sort of vasodilation is actually affecting even to the vessels in your brain. So typically, this is information that is considered a nuisance and try to get rid of it in our functional brain analysis. But I want to use it as a feature to understand how these pulsations that are not related to neural function, actually they may relate to your heart function and respiration. The way we did this is actually taking advantage of a really cool repository called the Human Connectome Project. It's already popped up in a couple of talks earlier, but this was funded to image with MRI 1,200 people. It was part of Obama's Brain Initiative, and just like the Human Genome Project aimed to crack the genetic code for humans, this Human Connectome Project has a huge database of brain images so we can map out all the structural and functional connections across all these different brain regions. And we have only really started to look at this only in a small slice of Human Connectome Project data. But I think what's really rich about the data is that we have this resting state fMRI, so the person doesn't have to do anything. They're lying there and we're looking at its normal brain functional modulation signal over time. But these same people during the scan had peripheral measurements. So while they were lying in the scanner, they had a pulse ox to measure blood oxygenation. And also they wore a respiratory belt to measure their breathing. I'll mostly focus on the heart today. But what we did is we looked at classic networks of regions, looked at the brain signal, functional signal at rest, and asked the question, well, how does this relate to heart rate variability, the heartbeat interval over time? And we did this using wavelet transform coherence. It does coherence over time and frequency. It's a lot of jargon. It's essentially asking how these signals are in sync or not over time and over different frequencies. And I'll just put one individual's example here. The bright colors in yellow are frequencies, for instance, where heartbeat interval and the fMRI signal, your brain signal in your visual network, are in sync. So it's not at all frequencies. There's two frequencies, for instance, this band, where they are highly in sync. And also what's interesting is if you look over the time, this x-axis is the time that they're collecting the scan while the person's lying there. Even though there's these two bands of frequencies where your heartbeat interval and your brain signals seem to be in sync, it's not always the case. There's some time points in the scan where they are in sync and sometimes that they aren't, which to me is really fascinating because does that reflect different vigilance or arousal states? It could mean a lot of things that we need to look closer into. So analyzing across regions, what we showed that there is this consistent relationship. Your heartbeat interval and brain bold signal, functional MRI signal, are in sync in specific frequencies. And we saw this across many functional brain networks, and specifically the visual, somatomotor, and ventral attention networks were highly correlated. The last piece of evidence I want to share from this Human Connectome project is when we have these signals, we can not only look at if they are in sync or not, but we can look at something called phase, which is the relative lag between the two signals. So which one is coming first? And if we do that same analysis, I'm showing the same example here. From a mathematical perspective, we often will represent phase by these vectors, like how the angle of the vector looks like. And you might have to squint a little bit, but you can see the angles here are looking kind of different between the two frequency bands. And when we kind of dove a little deeper into it, so the default mode network that I mentioned earlier, this is the network that turns on when your mind is wandering. In the lower frequency of synchrony, the fMRI signal in the brain tends to lead or come before the heartbeat changes, whereas in the higher frequency, the heartbeat changes tends to come before the brain changes. We saw this in multiple brain regions. I'm just showing another example of a network here. But I think that's really cool because now we have a way mathematically to tell from our resting state scans by comparing, hey, which is coming first? Is it your brain signal preceding the heartbeat interval, or is it vice versa? I think this has a lot of implications in terms of like what we could study in the future, so different autonomic or sleep states, even some of previous talks on potential emotional regulation. You can imagine using methods like this, right? And trying to understand like why certain brain regions are more related to the heartbeat interval or not. Is it because of the vasculature architecture in the brain? And then finally, in the health-related aspect, this is important to understand because as we age, it's obviously not just our brain that changes. Our systemic changes in physiology, like your arteries in the periphery, also tend to get stiffer. And what kind of impact does that have through heartbeat on your brain functions? I think this is an avenue to study the brain and body link from a non-invasive imaging perspective. So I want to end with just a few forward-looking thoughts. And one is just the tremendous potential of what I already shared, which is quantitative vascular imaging of the brain. My colleagues and I have contributed to designing a protocol to add in, for instance, perfusion imaging, so directly looking at cerebral blood flow in the brain in large multi-site studies like the Alzheimer's Disease Neuroimaging Initiative. I think we need more of these type of metrics so that we can push MRI again and brain health toward a quantitative metric, just like controlling blood pressure for cardiac health. Maybe this is a way that we can learn to measure and set health targets for blood flow in the brain in the future. And we heard about this in the last talk too from Brian, but really, with all of this vascular perspective, lifestyle intervention is still the best way, I believe, to protect your brain health. So there are ongoing large studies, such as US Pointer, that look at these interventions, such as physical exercise, cognitive and social interactions. These interventions that aren't pharmacological, that are lifestyle interventions, I think can be paired with neuroimaging, like I've shared. And this will be a way, I think, for targeted lifestyle changes and follow-up to really understand how we can better our brain health in the long run. And the last thing I want to share is that we at UC Davis and our Alzheimer's Disease Research Center is really committed to diversity and healthy brain aging for all. Our center has 26% Black and 28% Hispanic representation in our cohort. And this is really important because these populations, for instance, there are racial ethnic disparities in terms of how vascular risk is present and how it affects your cognition, which we can help to understand through imaging. And to me, as an engineer, when I see this and know that a lot of populations may not be aware of MR, it tells me I need to make those methods accessible, and I need to educate people on what it means to get an MR and what kind of information you can get out of it. So to conclude, I have a vision and our lab is working toward maximizing quantification with MR, but specifically of brain vascular function. And our ultimate goal is early detection so we can enable lifestyle interventions and really healthy vascular brain aging for all. So with that, I really want to thank all my lab and contributors, you for your attention. I'd be very happy to take questions. Thank you.
Questions?
How would you say studying neurovasculature would affect diagnosis and understanding of tumors like glioblastomas and their recurrence?
Yeah, I think glioblastomas have very unique vasculature, and especially things like tumors and also arteriovenous malformations, they have very specific vascular characteristics. So I think these sort of metrics are key to understand, like if their blood flow is being shunted, what's happening with oxygen delivery. I think those are unique circumstances that we can monitor over time and understand those diseases better. Yeah, thanks for the question.
If the MRI test depends on the oxygenation of the blood, what is the best time of the day to get an MRI test to get a more reliable and realistic result?
That is a great question, and honestly, it's in terms of functional interpretation of resting state. I mean, whether having your eyes open or closed also makes a big difference. So I think that tells me in designing large-scale studies, you might want to be rigorous about recording time of day, but you're absolutely right. It varies a lot with time of day. But that could be an opportunity, for instance, to study sleep. So if you collect resting state scans during sleep, your glymphatic system, so these fluids that are pushed around and related to your vasculature, but not quite the blood, that has a lot of dynamics during your sleep. And it's thought to remove abnormal proteins that potentially contribute to neurodegeneration. So I'd love to be able to use like resting state as it changes during sleep, actually. So in that sense, the diurnal variations are actually good because it tells us about different processes. But yeah, I don't know that there is a particular time of day per se that I would target, but it is something to control for, yeah. Thanks for the question. Oh, there's another question.
I think for me, heart rate variability might be what MRI is like for you. But you know more than I do about all these things. Do you think that there's a place for heart rate variability in the near-term future being applied to clinical work, not just part of a research program like yours?
You mean measuring heart rate variability as like a clinical measure, or?
As an idea of whether or not you need to do more meditation, more of something, just a clinical measure.
Yeah, absolutely. I don't know if that fits immediately into my program, but researchers who do study mindfulness have shown actually increased heart rate variability with meditation and mindfulness training, and they have linked it to specific changes in functional networks. So within networks, but also between like the frontal parietal control network and the default mode network. So it's more being able to kind of regulate in some sense between those two networks. So people have done that and specifically looked at increased heart rate variability as an aspect of that. It hasn't been yet part of my research, but it's definitely of interest. Yeah.
So you showed these graphs where there were things synchronized at lower and higher frequency. What do these frequencies mean? What does a synchronizing at a low frequency mean versus a high frequency?
Yeah, that's a really good question. I mean, right now it's an observation of biologically where these synchronies happen. In a practical sense, like the lower frequency actually overlaps with the frequency that a lot of functional MRI experts use for resting state network analysis. So one thing is just to be cautious, especially the lower frequency band, there is still synchrony and contribution of heart rate. The higher one, I think, is a little bit newer and needs more interpretation, especially since with fast imaging, so resting state fMRI, it has become faster and faster too. That's how we can get these high frequencies in the first place. So it's a little bit from a methods perspective, that's interesting. We should keep going fast if we can, if we have the sufficient signal. But biologically, I mean, those bands do relate to the natural frequency bands of heart rate variability. But I don't know if there's additional biological underpinnings of that. It is the frequency bands of heart rate variability as well. Yeah.
Thank you so much, Dr. Fan. We're out of time.
Thank you.